Start recording
Okay.
or
I start. Welcome
So hi.
everybody to today's webinar. Thanks for joining to today's webinar. This webinar is the third session of a joint work between IEEE Norway Young Professionals Section and HVL Data Science Group. Today we have Dr. Silvio from the Federal University of Pernambuco in Brazil. But before Silvio starts his presentation, I would like to thank Dr. Ilker, who is the associate professor in our campus, for inviting Silvio for this webinar. And I would like to ask Dr. Ilker to introduce him to us. Ilker, the stage is yours.
All right, thank you, Mojtaba. Yes, as I have been introduced already, my name is Ilker Meric. I'm an associate professor at the Western Norway University of Applied Sciences. So today we have an very interesting talk by one of my good colleagues from Brazil, Dr. Silvio Melo. The title of the talk is Bioinspired Texture Mapping for 3D Microprinting of Self-Decontaminant Materials. So Silvio holds a PhD in computer science from the State University of Arizona and is now an associate professor at the Federal University of Pernambuco in Brazil. His research interests include computer-aided geometric design, tomographic reconstruction, computational mathematics, computer vision, and their applications, medical applications. So thank you, Silvio. It's nice to see you again, and I'll give the word to you.
Thank you, Ilka. It's nice to see you again also. And thank you all, you guys, for inviting me to talk about something that I really am enjoying now, to be part of this project. Before I start the talk about the project itself, I'd like to put some context for the general audience. The context number one is the outbreak of the COVID-19 pandemic that happens last year. And for us, the impact of this, among all the obvious impact, the impact of the demand for equipment, a lot of different types of equipment, for ventilators, respirators, the equipment for protection for the health professional. And so the demand for this equipment increased a lot. And so this one was a fact that impacted the area of material science, as we will see later on this speech. Another thing was that the 3D printing area was also part of the effort to combat the pandemic, because it was an easy way to produce this material, all these devices, very fast and easy to produce. And we will talk about this also a little bit later. So we have two fronts in here. We have the equipment need for the patients, the respiration devices, and also for the health professional, the individual protection equipment. And if you join this to the fact that in the hospital, we had a lot of people coming, in excess number of people because of the pandemic, there was a preoccupation about the higher risk of hospital infection. So there was a preoccupation to produce equipment that would be able to prevent a little bit from the hospital infection, if it's possible. So the context number two is about this professor, Professor Petrus Santa Cruz. Professor Santa Cruz is a chemist that works in the area of material science, and he has a lot of fronts in research. He has a lot of contribution, a lot of patents. He's a very active scientist. He has a paper that has like 1,200 citations. So he's very active, and he works with all these areas and many more. He works with additive manufacturing, with 3D microprinting, photopolymerizable nanocomposites, self-decontaminant surfaces, microneedles printed in DOD, drop-on-demand type of printer. He also works with needles printed in 3D for drug transdermic administration, contaminant detectors, bioinspired superhydrophobic surfaces, and many more. What we see here in this on the right is a kind of a leaf that presents the property of superhydrophobicity. What we have here is a drop of water on the surface of the leaf, and we can see that the water doesn't wet the leaf. So, and the reason for this is because of the material properties, the levels of micro and nano scales that can produce this. We'll talk about more about this, but this is something that one of the things that Professor Petrus already was working on. For example, here I talk about a little bit more about this leaf, which is a yam from the northeast of Brazil, and also occur in other countries. So what we see here is that the superhydrophobic property comes from the contribution of different scales, mainly the micro scale and the nano scale. The micro scale, we see what we call papillae, and the nanoscale, the wax crystals. And together they don't allow the drop of water to wet. It stays in the shape of a sphere. And the people in the area of Professor Santa Cruz also define this hydrophobicity through this angle, the wet angle, or the contact angle. If it is this angle measured from this point and goes all the way to the other side, if it is larger than 90 degrees, we say that the surface is hydrophobic. And so this is one point. Here we see the lotus leaf, which is another plant that presents this property of super hydrophobicity, and many more that we can see through the scanning electron microscopy. The lotus leaf and all the other plants, and the last one, the Colocasia esculenta, is the one that the Professor Santa Cruz works with. But Professor Santa Cruz also worked with different material properties, like the structural coloring. For example, in this butterfly, the color blue that we see is not because of the pigment, but is because of the topography of the surface in micro and nano level. They produce this color. And it is already being used by cosmetic industry for this pigment in the eye. But the self-cleaning properties here, self-cleaning property is what we call the superhydrophobicity. The lotus leaves present this. Also, the adhesive, dry adhesive produced from this suction that we can see in the leg of this gecko, and also the fibers, the nerve fibers also that is used for artificial nerve. So we have all these properties, that self-cleaning, mechanical properties, structural color, optical properties that we observe in the animals and plants, to the biological property. So Professor Santa Cruz, already before this outbreak of the pandemic, was already working with this problem, which he has these plants. He has this plant in his lab. So he used this AFM, the microscope. The microscope is able to get part of this leaf and produce an image. This image I'm gonna talk about a little bit later. Is able to from the image we can actually reproduce the topography, the 3D topography of the leaf, which is then sent to this software, Topo Slicer, produced from our group in the computer science department that can slice all these objects in order for the printer to print layer by layer. This printer is what we call DoD, or drop-on-demand type of printer. So, okay. So more or less what happens is this. You have, sorry, excuse me, I have something, excuse me.
Reza, Mojtaba, I wanted to ask you something, by the way. The recording is still paused. I don't know if that was supposed to be the case.
Sorry, guys, sorry.
Silvio, can you give us 2 seconds? I just wanted to ask Reza and Mojtaba about the recording.
Okay. Yeah, continue, Silvio.
Okay, thank you. So this thing that I'm talking about is important to understand the purpose of the project. So what we have here is like a AFM by the micros. Oh, sorry. Okay. So this is AFM image from atomic force microscopy. In this image, what is the gray tone is related to the height of the structure. So the lighter the color, then the higher the height. So this image is submitted to the software for the conversion. The idea is to from the image to produce several layers to send to the printer. So the printer will print the structure in 3D layer by layer. So what happens is you get this structure, which is darker is lower height and lighter is higher. Then the software will actually produce the layers by using this criteria. I mean, the first layer, for instance, will paint in black all the pixels that has the level 1 or higher, and then the next layer will be only for those pixels with 2 or higher, and then the next layer 3 or higher, and so on. So then we produce the layers in such a way that the layer in the higher level will be totally grounded by the layer underneath, which is important for the printer. And then the context number 3, which is important for us, is this pipeline. It's a pipeline of 3D printing. So in 3D printing, you have an object like this. You model the object in softwares, CAD softwares, computer-aided design software that probably use parametric type of surfaces like NURBS. But then you need to convert to triangles in this format called STL. The STL is a format that is around since the 80s. It's a very old, but it's still very much being used in the 3D industry. So, and we'll talk about this a little bit later. So basically, you have the applications. The applications basically the application that works with the object, the model in the computer by using CAD kind of software. And then when somebody wants to print this object, the system converts into STL, so it converts from these NURBS into STL. This has some loss in precision. If you want to keep the precision in the level that in the higher level, you need to increase the number of triangles. So we have sort of parameterizable number of triangles. The more you have, the more triangles you want to convert, the better the precision. Then the STL file is sent to the printer, where the printer is still uses some third-party tools to manipulate the object if necessary. For instance, some tools may take the object and reorient it in order to minimize the support material that we see in the printer, because the printer prints the layer by layer, putting the object itself and also the support material in order to structures that are bending, like in the air, will not fall down when you print the layer in the material that is not dry yet. So then the third-party tools will do all this adjustment, and then the printer will convert this into a PRN file, which is a particular type of file that is in the printer manufacturer. And then this would be the slicing, to take the slicing of the material, like in this picture we see here the object here, original, and then we have all in layers. In each layer, the printer will do some pattern of material deposition in order to make the object stronger. So basically, this is the key point in the pipeline of the 3D printing, is this slicing of the object. Then after the slice, then the 3D printed, and then after that, we have the post-processing of the object, because you may have to do some cleaning or remove some support object that is still attached to the object. And then this is the whole pipeline. So the key point here is this STL file. As you see in this object here, we employ a lot of triangles. You can see here, and this can be a problem, because the number of triangles, if it is a very high number, like in millions of billions, then probably you not print in due time. And this is something that we have to deal in our project, because in our project we have a miniaturization of the object, which require even more triangles. As we see here in this venturi valve, the amount of triangles here, you can see the details here. Actually, if I can show, can you see here the venturi valve as we move here? This object has like almost 3 million triangles. We can, in this software, we can actually show it closer, and then we can see all the triangles here. The amount of triangles is really high in this case. So the reason the industry still use STL is because of this slicing. This slicing is basically, you compute the intersection between a plane. This blue plane is parallel to the printing bay. So what we do, we compute the intersection of each triangle with the plane. which is an edge, with this edge here, is the result of the intersection. I have actually something that can illustrate this better. You see here the software GeoGebra showing several triangles being sliced. I mean, this is like a layer of triangles being sliced by this plane. The result is these edges, these red edges here. So the intersection between a triangle and a plane is considered very easy, very fast. So that's the reason why they still keep this standard. By the way, we in our group in here, we worked with a project in 2018 and 2019 that was funded by the HP, the Hewlett-Packard, that produce 3D printers. And we worked with a project that was basically to improve this standard. We were actually working with these NURBS type of surface. NURBS is the acronym for non-uniform rational B-splines. It's a spline type of surface. So our project was to do the slicing directly on the NURBS instead of doing this conversion to STL before. So this is our project, was our project. And we did this successfully, but the industry hasn't yet adopted this because all the software is still, people are still producing software that work with STL. So is it the force of the industry that doesn't allow to go further in this standard as we speak, as of this year at least. But in the next years, I believe we're going to change for the parametric standard. The NURBS is a parametric standard. So the other context that is necessary for us to understand the project is the texture mapping. Texture mapping is a phase in the computer graphics pipeline. That is basically you get an object, you take an object, and you also will take an image, we call the texture, and then we produce the object with the image applied on the surface of the object. So there are several techniques for doing that, several very sophisticated techniques. For instance, this image here, what we have here, these bricks, they are not real bricks. They're just texture applied on the flat surface. So we have this illusion of something 3D. So the texture mapping is something really important in computer graphics. We have all this effect that we call flat object that has no texture, smooth texture, and we produce this type of synthesis of image. But in our project, we are interested in a particular type of texture mapping, which is the following. We have the object, which is STL object. So the object is all made of triangles, flat triangles. And each triangle has a norm, the vector, normal vector, which is the vector that is associated to each vertex, instead of the plane itself. Because in computer graphics, we use the neighbor of the triangles in order to determine which direction the normal vector should have. This is like you want to more or less to model the original smoothness of the surface. It is as if you wanted to go back to the NURBS surface from the triangles. So it's like something smooth that was switched by, they changed by the triangles in the conversion. Oh, sorry. in the conversion, or something like that. You change by these triangles. Each triangle, each plane of the triangle has its own vector, normal vector. But then, since you want to model the smoothness of the surface, you want to actually let's give back a little bit of the smoothness. So we associate the normal vector to each vertex instead of the face. What we do is we take all these normal vectors per face and we do the average. We average all these normal vectors of the triangles that has this vertex in common. Then you associate this average of these normal vectors to the vertex. So that's why in this drawing we have these normal vectors associated to the vertex. So what we do is our interest is to actually do a texture mapping using 3D information. So information that can change the height of these triangles or the position of this triangle in the direction of the normal vector. So when we get the texture, when we take the image that we call texture, we actually, each pixel here has the height, has the information on height. So we take this information and we add to the vertex of the triangle that fall here associated to this position. We just add it to the height. So we modify the triangle's geometry. So it's something like this that happens. We have this sphere, and then we modify the sphere to the geometry of this sphere. This sphere is actually modeled through triangles here, several triangles. And then we modify the position of this vertex in order to present this new geometry. So we actually modify the 3D geometry of the triangles. So last year, the CAPES, which is one of the research funding agencies in Brazil, presented this call for this. The idea was to help in the pandemic effort. So the idea of Professor Santa Cruz was to present a project that was whose purpose was to recover the equipment, the devices, the respirator and the ventilators with these these patterns or these topographies, in micro scale and nano scale in the equipment in such a way that the equipment will be self-decontaminant. I mean, it's like it will repel bacteria fixation. So the importance of this is that while people are trying to protect from COVID, it is nice not to have the cross infection from other types of microbes, like bacteria, for instance. So this was the idea. So the name of the project was Interdisciplinary Network for the Development of Self-Decontaminant Materials with Active-Passive Hierarchical Structure aimed at fighting the COVID-19 pandemic and other outbreaks. By active refers to chemicals with bactericide effects. So the chemical itself can kill bacteria. And passive refers to the microtopography or even nanotopography on the material surface that has this property of also repelling, at least repelling bacteria, but also can kill bacteria. So this was a way to subdivide the project in groups. So our group was responsible. By our group, I mean the computer science group to have 2 professors and several students from the computer science, and also Professor Santa Cruz has several students from the material science postgraduate program. and also some other professors working with him in the chemistry department. So then our part in the project became this texture mapping. So we get the image, the AFM image of a biological structure known for being self-cleaning or self-decontaminant or even super hydrophobic. And then we take this image, we construct a displacement map from the texture. Displacement map is just the information of height itself. And then we do the displacement on the surface itself to be printed in the printer, in the 3D printer. So that's this is our part in the project. The other part of the project, the active, I mean, this part we call the passive part of the project, which is the self-decontaminant property comes from just the topography of the surface in the micro and nano scale. But also the self-decontaminant may come from the active part, chemical part. So we are working in parallel in these two fronts, the active and the passive structure. So this is like a summary of the project. So we have these chemically active systems, which is Professor Santa Cruz and colleagues, to work in this subject here, photopolymerizables nanocomposites. He works with copper and silver nanoparticles, functionalized surfaces for photodynamic action. We will end up producing these micro needles, 3D printed with active materials. This is a front that he works with his student, is to administer, to produce the drugs and to administer through these micro needles in the skin. And also he can use the same idea, but much smaller scale, nano needles, printed in drop-on-demand type of printer to pierce the protein envelope of viruses. It's something more advanced that he is working on. And our part is this chemically passive systems. And we have basically we have two lines, the Bioinspired topographies for reduced adhesion of bacteria and Bioinspired topography for increased adhesion of bacteria. So we're going to use both. Reduced is obvious, but why increase adhesion of bacteria? Because in case the topography is capable of piercing the protein layer of the bacteria, then it's important for you to keep the bacteria fixed in this topography in order for the rupture to occur. So then we work on these two fronts. And then we have a different part of the project, which is more related to detecting the contamination, the efficiency. There are several systems for just detecting the contamination. So then we talked about our part in the project, these bioinspired topographies for adhesion of bacteria. So basically, we have this front, which is directly related to the texture mapping. We call bioinspired texture mapping because we're gonna get the image from biology structures are already known to present the property that we want, the self-decontaminant property. And then we do this texture mapping, as I explained earlier, by adding to the vertex these heights coming from the image, converted from the image. So we modify the structure to present the same micro and perhaps nano structures that we see in the biological structures. So this in computer graphics, this is not new. It is not innovative. But there's a challenge here. The challenge is to deal with an explosive number of triangles, because this thing is miniaturized, needs to be miniaturized in order for you to apply the texture, very small texture in micro and nano scale. For instance, here we have part of this venturi valve, and we see all the triangulation here. And then the actual size is 5.57 by 2.78 millimeters. We have all these, this is the size of this whole rectangle, and we have all these triangles you see here. Just to have an idea of the size of this triangulation, comparing with this pencil, with this 5 millimeters pencil, we see it's very, very, very small. And then this is an image from AFM, the microscope, atomic force microscope, from this Colocasia esculenta leaf. This is the image of in the micro scale of this leaf. And we see here a triangle, an actual triangle of this size, the size of the, like, the size of something like this, triangle like this. This triangle I put on the image to show the actual size compared to the structure. So, and then remembering that when you apply the texture, you only can manipulate the vertex of a triangle. Only. That's the only place where you can modify the height of the triangles on the surface. So you see that this resolution is very poor. So we need to retriangulate even more. So we have to create new triangles. This, for instance, is a triangle subdivided into 63 triangles, new triangles. It is more or less the resolution of a 3D printer of today. It's like something like 16 micrometers or 16, 20 micrometers. But it's still growing. Maybe we can find already new printers that can go even smaller than this. But then, so this is the kind of resolution that we get with the current printers. So you see that it is still a poor resolution, even with this subdivision, it's still poor, because we can see that we will not be able to reproduce all these nice, structure here, because, for instance, we have something like this. You cannot show the interior. You only you take the height of each vertex here is where we can show the height. So the change in height in this interior will not be shown in this triangulation. Here is a tetrahedron with a triangle in a size more or less compatible with the printer, each face. Then let me, excuse me, let me show you the actual. So can you see this tetrahedron? So this is a tetrahedron with the texture already mapped. Let me put in here with some illumination so you can see more or less the structure in micro scale and nano scale. But this is something that we can do in the software, but not in the printer. Printer cannot get in this resolution. The printer is poorer than this. So in our part, in our project, where we come from, the computer science part, we are free to try arbitrary resolution, because we know that the printer are each year getting much better with the resolution. So it's still a good idea to attempt to do this kind of texture mapping. But the problem is still a huge problem: how to work with an explosion of triangles. So we have something like two ideas. One of them is to apply texture during slicing. So it's something like this. This image shows what we are thinking doing. We have this slicing of this layer of triangles, and then what we do is we do the intersection between a given triangle with the plane. Let's say this triangle. Then we identify where this line, where the triangle will fall on the texture mapping, the texture, the corresponding place of each vertex in the texture mapping. We identify where the line will fall, this line of intersection, and then we get the cut of this structure. We see the variation of the height only at this line, on this line. And then we change the slice. They change the edge. We cut the edge in several parts, and then we change the height on the edge only, instead of retriangulating and then computing the height for each vertex. So this is a way of not multiplying the number of triangles, because you can imagine something like this, or something like the Venturi valve. The Venturi valve is like 2.9 million triangles. It can get easily to the billion of triangles, easily, if we do the retriangulation using the current printer resolution. So in order to avoid these billion triangles, we only take each triangle and we do the intersection. We take this line, the edge, we see where the edge will fall in the texture, and then we see the variation of the height. Then we change the position of this layer, of this edge on the layer. This is one idea. The other idea is we can actually do the retriangulation, work with the billion triangles, but then we can store as in a database, but we store in such a way that we can load into the memory layer by layer. So we only load a certain layer of triangles that correspond to a plane, something like that. Some structure that we can, some data structure that we can that it allows us to take only a layer of triangles. So this is something that we are working on. This is the first line of research. The other line of research is the simulation of bacterial adhesion to rough surfaces. So it's something that we want to produce the topographies in micro and nano scales to be more tailored to our application, because it is known that bacterial adhesion is a very complex system, and is affected by different variables that can be like electric type of forces, also chemical, and then the topography also. So we want to actually to do the simulation while we experiment with different type of topographies, and we see the results, because doing that in the actual experiments is very difficult. We saw in the literature that people have a lot of difficulty to produce experiments that can test different type of topographies. They only can do some very simple experiments, very simple experiments with topography, different topography. So we thought that we can work in two different lines. We assigned this to a PhD student, the bacteria in planktonic state. Planktonic state is the isolated bacteria, in contrast to biofilms. Biofilms where the bacteria form like a community, where they interact with each other and form a strong bond that makes difficult to remove the bacteria. So for instance, we can see here a few type of topographies. We can see that, for instance, the cicada wings has a sort of self-decontaminating for certain types of bacteria. And it's basically made of nanopillars. And these nanopillars can rupture the bacteria layer, the bacteria membrane, as you can see here in this picture here. The bacteria being pierced by these nanopillars. But these are only just one type of bacteria. This is like it's called, this name here, something like aeruginosa. Pseudomonas aeruginosa. But there are several types of other bacteria that will not have this problem here. And then you have the experiment with different types of topographies. We have this type of topography here, where also there's a bacteria that is killed by this type of topography. We have something also like this, that the nanopillars can kill certain types of bacteria, but other types can actually adapt to the nanopillars. This bacteria, this green was painted by green just to make it easier to see. The bacteria like assume this orientation, so it will not be killed by the nanopillars. So this is something that motivates us to attempt producing new different types of topographies in micro and nano scale. This is something, it's like an illustration that show how the rupture happened. There's some work, this guy is Alborn, that you all describe this in details. And then we have this article that came up in 2021 that brings the simulation that we are thinking working on. They simulated bacteria as like a cylinder with these rounded ends. And then here in this figure, it is showing the contact between the bacteria and the different type of topography. The two different type of topography. Here we have a bigger, smaller contact, you have a bigger contact here, depending on the topography. The contact is like a way to measure the force needed to attach the bacteria to the surface. Now is some details in this paper that show how they model the bacteria. They model as triangles, a lot of triangles. And they also show how to produce the interaction, the forces of interaction between bacteria and the surfaces, how the force affect these triangles. They show in the paper how this is done, how each vertex can move, how the vertex will deform the form of the bacteria in two different types of bacteria. And then they simulate with the surface, the topography, only these two types of topography they experiment. They call these lips, this type of topography, and these they call the nanopillars, which is it's very, I think it's very modest as to call these nanopillars. But I mean, this is what they experiment because of the difficulties of this process. And they also they study, for instance, when the contact between the bacteria and the surface and the stress and the forces that are produced in the interaction between the bacteria and the surface. This is the type of thing that they study. So in our case, we want to do something similar to that, but we want to be able to parameterize this topography a little bit better, a little bit more control, but also want to experiment with this totally different type of modeling, this this kind of physical situation. It's like using this multi-particle hydrodynamics that we believe is better than the finite element that they use here, because of this SPH is better to model the interaction between two different surfaces. For instance, here this wall is totally made of particles, and they put some limitations or some constraints on the way the particle should move, so the particle stays static here. They do this through forces, so it's a natural thing for you to model two totally different surfaces by using just adjusting the particle, the property of the particle. They can actually simulate that equation, the Navier-Stokes. Navier-Stokes, probably you know this Navier-Stokes, that is able to model different phenomena like flow, liquid, and all different type of phenomena, natural phenomena. So we can simulate the Navier-Stokes, like you can adjust, for instance, the viscosity. You can adjust several variables. So we believe we can model the bacteria by using particles to model the bacteria, and also to model the surface, the surface itself, the topography, and also the substrate, the liquid where the bacteria is floating. So we have this line here. And the second line in this simulation is the bacteria in biofilm. We have a student. We have a student that is working on this. So biofilm is a totally different system that appears when a lot of bacteria, after a time passing, if you let the time pass, then we will start to accumulate bacteria. And then after that, it construct a community. This community.
Silvio, we have 3 minutes left.
Oh, sorry.
For your information.
No, this is basically, it's almost the end.
Okay.
So we believe that we can simulate also this the interaction between the biofilm and the topography, because there are several things that happen that is different from the bacteria in planktonic state. For instance, when you have a lot of bacteria, these bacteria in this part are the pressure is higher, so the bacteria is really pressed, and the system here is lower for the bacteria, for the food that comes to the bacteria, and a lot of other things that happens is lower, and also the pressure may produce interaction that we can actually combat with a certain type of topography. So this is the idea, to be able to produce a topography tailored to avoid the biofilm formation. So this is basically what I wanted to say. I hope you understood my English. So we have questions?
Yeah, thank you so much, Silvio, for your great presentation. It was really fascinating for me. I really enjoyed how material topology, can kill bacteria or stop their activities.
Thank you.
Yeah, I think Ilker, do you have some question?
All right. Thank you, Silvio.
Mm-hmm.
It is totally outside my field, but anyway, I'm wondering something. So maybe. So you're looking mostly into the topography itself when it comes to the adhesion rates, let's say, right? I mean, that's what you're concerned with. But what I'm wondering is whether there are other parameters that would affect the adhesion rates, such as, you know, the temperature, humidity, I don't know.
Yeah, a lot of,
besides that,
actually a lot of parameters. In this work, this previous work, let's, I mean, I think, no, I think it's, sorry. What's the name of the guy? This Elborn, this guy. This work has is a very nice work. Has a lot the details that we know so far about this bacterial interaction with the surface, and has a lot of things. There's a lot of variables that affect the adhesion. But the ones that the variables that are most important has to do with the topography, and also some type of a chemical system, some chemical system that happens with bacteria. And also it's complex to say how, at this point at least, how the topography will be successful in general, because what we observe is that certain topography is good to avoid the adhesion of certain type of bacteria, but to other type of bacteria it's totally ineffective. And also we have a difficulty with the technology of printing devices, that because what we know is that the nanostructure and nanoscale is the one that most affect this interaction, and we are not yet capable of printing in nanoscale, at least with 3D printers. So we're still waiting to see the technology to get better in order for us to apply the knowledge that will be produced from this simulation. But I wanted to show you. Let's see here. This biofilm. Excuse me. Let me just take airborne bacterial nanostructure. This is the article. Bacterial nanostructure interaction. So it has a lot of things. For instance, you have two types of bacteria, gram-negative and gram-positive. This is something that changed totally the interactions. And in this paper, it says that the hydrophobicity or hydrophilicity, they can both contribute or can stop the adhesion. It depends on several factors. So it's very complex. We still are in the beginning of these studies. So a lot of things a lot of work to do.
Yeah. Then that brings me to the next question that I had, and that was, let me see. As you said, I mean, you're not able to print yet using 3D printers at the nanoscale, right? That's we're not there.
Yeah.
Have you considered looking into lithography, maybe, which is done, for example, for nano coating and so on and so forth? It's commonly used in nanotechnology, at least. So.
Okay. I believe
electron beam lithography and so on and so forth. I don't know. I mean, your challenge is basically that you need to print larger objects, right? So this is not basically lithography.
Okay.
It is not for that as such, but it could give you some inspiration, maybe. Not sure, or maybe a different angle. That's fine. I don't know. Have you considered that or?
I believe the Professor Santa Cruz has considered that from our meetings that we had. He had all these different line of research. He has two labs. He coordinates two labs, and one of them we have actually the sorry, the logo of the lab is, where is it? It's this nano ponto quântico nanotechnology. And I'm sure he experimented all these things. We have several different technology being experimented here in this lab, ponto quântico nanotechnology. But I'm not, I don't know much about this, actually.
No, that's fine. Then one final question. So if this research is successful, I mean, what do you think its overall impact will be? Just curious. So.
Okay. Yeah, I think it will produce equipment, I mean, surfaces that will be probably will be really used in different type of equipment for hospitals because of this. Because if it is successful, if all this line of research get to this point where we hope we get, it will be a very versatile type of surfaces, because in terms of avoiding bacterial infection. For instance, the biofilm formation, it can avoid the biofilm formation, and for a number of, at least for the types of bacteria most commonly seen in hospitals, at least. So this is our hope. There will be, I mean, for the industry, it will be very important. But we will, I mean, cover basically all types of devices. We see that some devices, mainly in the inner part of the device. If you take here, sorry, just here the venturi have some inner part here, inner. I don't know if you can see it. And this is actually where the air has the interaction, big interaction with the device. The air pass through these holes here, these structures, these pipes. So this is where the recovering of this surface you benefit the most, because this is where the bacteria used to accumulate, I mean, to form biofilms. So we imagine that these devices, instead of, because what happened today, we have to throw these devices, I don't know, perhaps a few weeks or something, or a few days, I don't know. But then with the new coating, then we probably will have this device to have to be able to use for much longer times, much longer period in the hospital. There'll be safer devices.
All
so
right.
The idea also is the hope. I think it's just a dream of Professor Santa Cruz is to be able to produce topography and also the active chemicals to avoid the viral contamination, which is much harder because virus is order of magnitude smaller than bacteria. But it's still, those nano needles that I talked about, that intends to be printed in DOD, those nano needles, the intention is to pierce the viral, I mean, it's not exactly pierce, it's like entangle the viral, those, I don't know, those proteins, those spicula, I don't know, spicula in the viral, it can entangle with these nano needles, and it will cause the death of the virus. the virus. You know what I'm saying? So this is the dream of the professor, but it's much harder because it's much smaller. It's like a bacteria is like 120 nanometers, and a virus is like order of magnitude smaller than this.
All right. Thank you, Silvio, and good luck with the research. So,
Thank you very much.
Yeah, thank you, Ilker and Silvio, for your attending to this webinar. And yeah, have a good day. It was a great presentation.
Thank you very much. Thank you very much.
thank you. Bye bye.
Bye.